core button.
There you go. Take it away.
Okay, welcome everyone. Good evening, people that join us from the BMC. Hopefully, more are coming in. Um, I am very excited to introduce Maria Alice Neves. I hope I said your last name correct, Maria. Um, if I didn't, I apologize. I came
It's
across
correct.
Maria. Yes.
It's correct.
It's correct. Okay. I came across Maria when I was reading a book, Let's Become Fungal by Yasmin, and I forgot her last name. It's all about how the fungi can teach us how to cooperate and how not to be afraid of death and decomposing and things like that. Because it just, if unless something dies, nothing else new can regenerate and start growing again. And that was something that you talked about in a book. That's what I read, your input in a book, and that was very exciting to me. And then I realized that you had a lecture about how the mushrooms affected and transformed the whole planet Earth, the development of the Earth, and how they contributed to the what we have now on planet Earth, right? So I'm going to read the little introduction about Maria. Maria is a professor at the Federal University of Santa Catarina, and you teach there basic and applied mycology. You're curator of the Florafungarium, and you collaborate on a toxicological center in Santa Catarina, identifying cases of mushroom poisonings. Hopefully not too many. You work on the mushroom identification and you help form the students at the graduate and undergraduate levels. You also study tropical ectomycorrhiza that you mentioned a little briefly to us. Maybe you will talk about it more. And you lead a group that investigates the partners that are native to the Atlantic forest in South America, right? You said that in the tropical area. And
Mhm.
you also work well, obviously you work in academia and you're promoting the research and outreach projects, including mycotourism and the art inspired by natural history. Hopefully not just fungi, but other stuff too. So go ahead, Maria. You can start.
All right. Okay. Thank you so much. I'm gonna start sharing my window.
Mhm.
Um, Give
Oh,
me
while
just
you
a
do that,
minute.
I'm just going to say
Yep.
if anyone has any questions, you can put them up in the chat and I can read them out, or maybe you can just unmute yourself since it's a small group right now. But I'm going to have the chat opened up so I can see if anyone asks
Uh,
anything.
so I'm just gonna might create something weird first, but
Oh,
let
great.
me move. I'll be moving you out of this for just in
Mhm.
a minute. Hmm. Oh, okay. Um, oh, let me just
Maria, I find that whenever you're
I
going to
thought
screen share, it works better if you open it and then hit screen share. If you get like open on your desktop and then hit screen share.
okay, so I'll stop share because I had it open. I don't know what happened.
Mm-hmm.
It's like it's like we did this so much during the pandemic and didn't learn anything about it. Forgot, blocked all the things that I did so much in pandemic. Okay, here it is. Okay, now I'm gonna come back here. Okay, how's that? Does that work now?
Yes, now we see it. We
Okay.
see your initial screen, Ancient Fungi. Where'd you come up with that title?
Yeah, so the title came from David. But I loved it. I think it's great. So this is I want to start just saying that this is not my original work, but it's something that I put together like from several articles and works from other amazing people and mycologists that had done that. And I put it together for start first for myself to teach about this, about how the fungi impact the evolution of Earth. And then it became like a desire, and other people started asking me about it. And then well, I'm telling you everything about. So my name is Maria Alice Neves, like Jean already said, and I want to thank you very much for the invitation. So this is not my original work, and just to start, I want to show where I'm based at, and I'm here in Florianopolis. That's the name of the city I live in. And it's an island off the coast of the state of Santa Catarina. So this is the island's about 425 square kilometers, or 265, about 264 square miles. And there are about 470 inhabitants only in the island, because Florianopolis has also like a continental park, but this is the population, approximately population that lives only in the island. Although it's very touristy in the summer, and in between December and February, the population of the island might reach like 1 million people or more. So it gets really, really crowded. And right now it's winter, it's pretty cold. It's about like 60 Fahrenheit tonight. And at this time of the year, we have visitors from the south, like the northern whale, some penguins, and schools of mullets that are like a very traditional dish in the island at this time of the year. And so, speaking of, art and how I started in mushrooms, I have been reading and I have been sorry?
How mushrooms impact growth on Earth.
Sorry? Is
So-
there a question?
Somebody's got their sound on and they're talking in the background, so I'll mute everybody, but
Okay.
can
So
everybody just mute
I
themselves?
started like I became interested in fungi during my undergrad. That was about 30 years ago. And since then, fungi have become like my object of study and research, and also part of, what moves my life in a way. And like what's in the book that Jenna mentioned before, Let's Become Fungal, fungi have taught me a lot, like how to collaborate, how to be more welcoming to diversity, and how to accept weird, because there's nothing more weird than the shapes and the colors and everything that we see on mushrooms. So I was very, very happy to be part of these interviews that Yasmin did. And I love to combine the science that I do and the natural history with stuff that I like to do, like sewing and embroidering. And this is an embroidery that I do, that I use in my classes a lot, that shows like the main 5 phyla of fungi, including like the aquatic fungi, the Chytridiomycota, the fungi that grow like on like more like sugar, like simple compounds, like the Zygomycota, the Glomeromycota, that are symbiotic with root of plants, the Ascomycota and the Basidiomycota, that are like more well known because they form the macrofungi, and other things that are like more used. And I know you all know how fungi are important for soil nutrition, because they're basic decomposers. So they decompose, they feed themselves by throwing enzymes on wherever they are growing, because like I say, their house is also their food. So they through the enzymes and then absorb everything that's important or interesting for them, but they decompose more than what they absorb, and that's how they feed also the forest and the soil, because all the compounds that are decomposed but are not used by the fungi are also uh, available to other plants and other nutrients and other organisms. Um, and the other thing that's always fascinated me about fungi is how they interact with all groups of organisms. So we have like, basic decomposers like on wood and the leaf litter, and on that animals like the cordyceps that decompose and attack animals, on live animals also. The lichens, like the fungi that are associated with algae or cyanobacteria, and also fungi that are used by other animals. Like this is a bird that is in the southern Brazil that collects the whole nest. You see how the nest is black? This bird collects mycelium cords from marasmius to build their nests, and it's really beautiful. And the same mycelium cords are used by the indigenous people in the Amazon, the Yanomami women that use the same cord to weave baskets. So fungi are like everywhere, literally. And even though we know how important they are and all the impact that they have now in our lives, also with food and fermentation, we know they are like the foundation of life. They're like so important for everything that we do, and they're so great, but they are never shown like in some parts even of science, or like they're very like little represented. And so this talks about how the fungi impacted the earth over like the last millions of years. And then when we look for that and we look like for these charts, because we can find several of these charts that show like the major events that and how these events change during like different eras, and there's rarely, I have never found one that included the fungi. So we have here like, it's a nice chart and it has like animals and plants, but do you see any fungi here? And when we think about like the Jurassic, for example, do you see any fungi there, or any fungi among the dinosaurs or among like the tree ferns? And normally we don't, and it's not something that we generally think about. The fauna is there, the flora is there, but the fungus lacking. And it's not because it's not there. So because of that, I started like doing something by myself, like just putting some arrows, like to have a class that represented that, to show to my students. And then I joined with Samara Antero, who is an artist from Rio de Janeiro. And we put together this chart that includes fungi in several spots. And so that's what I'm gonna talk about it today. So this, we have done that in English also. So with this QR code, you can download this, I'll give you some time. You can download this exactly chart that is here. And then at the end of the talk, I'll show the chart again with some points where the fungi are shown, so we can see like how many times like fungi are have impacted the earth. And that's, so like I said before, this was compiled from articles from several different people, several amazing mycologists that have worked separately in several fossils, but I had never seen like this all put together in one chart. And we did that, and we also wrote in Portuguese for now a little booklet that talks about the history of the fungi and the evolution of Earth, including the chart, but also a text that I worked with one of my undergrad students, Miriam Sprisigo. And it's written in a language that's like for anybody. Our goal was mostly like teachers, school teachers, and anybody that is like interested in, natural history and fungi. So this QR code is for the whole booklet in Portuguese, and I intend to translate it soon to English also. So we are starting here. I have like this chart, the same chart in miniature, so we can follow where things are happening. And here we are in the Precambrian, in the Ion Proterozoic. So this is like about 2.4 billion years ago. And this is a very ancient fungi or fungus called Orosphida giraldi. And this fungus is, one of the hypotheses is that it was associated with other organisms that were also in the Proterozoic at the time. And one of the hypotheses is that was in symbiosis with some other organism that was already like also in Earth at that time. So that shows how old, not only how old fungi are in terms of geological times, but also that symbiosis with other organisms has always been like something that was natural for fungi. And this is the one that Jana was saying that she remembers. This is the genus is called Prototaxitis or Prototaxites, I would say. And these fossils were found in the Silurian, so much later here, about 440 million years ago. And these were huge columns formed by mycelium. They could have like about 29.5 feet tall. And here we have like a paleontologist with one of these fossils. And there is a beautiful article by Marc-André Celos from 2002 that talks about the hypothesis and how they are formed based on the studies of the fossils. So they were like big columns of mycelium, but there were other organisms growing on top or in symbiosis of it, like some algae, especially. And one of the questions that is not heard, but one of the hypotheses that they would maybe be huge columns of lichens. So again, we see the association with other organisms. So this was like a common theme in the Silurian. Then this to me is one of the most impactful events that we have evidence in the Devonian, because in the Devonian about 410 million years ago, the basic plants like in the genus Aglaophyton started moving from the water and started to grow on land. And these are fossils of this genus, Aglaophyton, which is a plant that used to grow in the Rhynie Chert in Scotland. And at the basal cells of these plants, they found these images of structures that are hyphae with spores that are very similar to what we know today as the arbuscular mycorrhizae in the glomeromycota. So the hypothesis is that these fungi were associated to these basal cells, and they were very important in the fact that the plants started to grow on land. So because we know the hyphae can grow much like in a much wider substrate, and they were able to then collect nutrients and water for these plants that didn't even have real roots yet. And this is like the base of everything that is started forming in the earth as a mycorrhizae, this big association that is like everywhere we know. So some facts that we have today that support the hypothesis that the mushrooms started the mycorrhizae and help plants grow on land is that more than 90% of the plants have mycelium or fungi in their roots. And some of these plants expand or give to their fungi sometimes up to 10% of the carbon that they produce from photosynthesis. So this is a lot. It's a large investment, which suggests that this is really an old, ancient relationship. And I'm gonna move to some like actual extant examples that I'm sure you guys are all like know about it. So here we have like pine trees that we know are associated with russula, species of russula, that the mycelium grows on the pine trees and form the mycorrhizae, and the pine trees do the photosynthesis and send carbon to the mycelium of the russula, because we know that fungi don't do photosynthesis, so they don't have their own source of carbon. And in contrast, fungi can expand the the area that the roots absorb water and amino acids and nutrients, and also exchange chemical signals with these plants. So this is like a one-by-one root or relationship that is everywhere. Like there are tons of examples of plants and fungi that have this one-by-one mycelium root symbiosis. We also know that there are several species of plants that can have, one species can have like several species of fungi growing on their roots. Um, and then we have this other example that I find fascinating, that it's the same relationship. So here we have a russula, the pine. It's the same story that I already told you, but there is a third element that is monotropa, or the Indian pipe plant, that is an ericaceae that do not do photosynthesis. And monotropa is also linked on their roots with the same mycelium of the russula. So what happens is that russula absorbs carbon, receives carbon from the pine tree, and transfers to the monotropa. So monotropa doesn't do photosynthesis, but needs carbon, and it gets that from the pine tree through the russula mycelium. So to me, this is fascinating. It's really cool. And there are also several examples. Another example is like a different species of monotropa that grows in Costa Rica, that instead of being white is completely red. And it also lacks chlorophyll, sorry. And it's associated with a species of Tylopilus in Costa Rica, which is associated to another ericaceae that is in the genus Comarostaphylis. So several of these examples that we know, we have facts and we have evidence, but these relationships are really hard to study, and I think they are probably more common that we know. We just need more studies and more money being invested in fungi research. And then they go one step ahead, and I love this. This is a study that was made with collembola, which are little Arthropods are insects that live on the soil and they feed on mycelium. So they were studying like how different communities of collembola, or these little insects, were related to different mycelium species of different fungi. And so the expectation was that they would feed on mycelium and the community would grow more or less. But when they tested with mycelium of Lacaria lacata, the community decreased and the collembola were dying. So they did this test where they put like the plant and the collembola with the mycelium in the middle, and the mycelium could connect to them both. And they noticed that the fungus or the mycelium was attacking the collembola and feeding and using nitrogen that was being absorbed by the mycelium, and they marked the nitrogen, and they recovered the animal nitrogen in the pine trees that were also associated by the same mycelium. So this is fascinating to me. So we have here like an example of how we cannot just simply put fungi in one box. So Lacaria clata is, yeah, ectomycorrhizae, but it's also a predator of collembola, and feeds off the animal nitrogen from the collembola and passes that through the mycelium to the pine trees. So we have all these beautiful examples of what happens like beneath our feet. And we need more studies to know more and to have more evidence. And with that, speaking of mycorrhizae, I think you might have heard like the common hyphae or the wood wide web. And we still don't have evidence to say that all the plants are all connected and all the forests are connected by the mycelium. We can't prove, but we can't say we have evidence for that like everything is connected. So it's probably not a free Wi-Fi for everybody, but we have evidence of a lot of examples of Wi-Fi with password protected, where there are some species that we know are connected. So there is a lot to be discovered and a lot yet to be studied in relation to the what's happening beneath our feet. So now going back to our chart, we jump to the Devonian. So those trees, those plants that started growing on land, started to grow. They had food, they established on land, and they started to grow. So they needed materials that would help them stay upright. And the first material was the cellulose. So they start to grow, they have cellulose now, so they can grow bigger. And the cellulose starts to be a problem because who is going to decompose the cellulose? So the fungi came to rescue, and we have then the appearance in the Devonian of fungi that have cellulose to decompose this new compound, which the fungi that we call as brown rot. So brown rot is called because the wood have disappearance when the cellulose is decomposed, but the lignin is not. We have this wood that is brown, it's dark colored, and it's like all kind of square, almost like if somebody cut squares out of it. But the lignin started to be built also, and the lignin was started to be a problem. And then come the fungi. In the Permian and the Carboniferous, the fungi that had cellulose, and some of them started to also produce ligninases to decompose all of the wood, to completely decompose the wood. So what was happening is that all that lignin for several years start to accumulate. So we had like piles and piles of trees, and that's what we would have today if we didn't have the fungi to decompose the lignin. We would have piles of wood. And that was what formed the coal depositions in the Carboniferous. So the fungi came to the save again, and they started decompose not only the cellulose and the lignin, and these are what we call the white rots. And they're called white rots because the wood becomes like very flaky and white, and it's a result of the decomposition, complete decomposition of the wood. So we can blame fungi on like stopping the production of petroleum in the world or the coals. And now we got like to the Jurassic, like the Jurassic Park, very famous movie. And the Jurassic is like what everybody think. I think the majority of people think of Jurassic when we talk um about like the eras and like ancient Earth. So we know we have like all the dinosaurs and the gigantic, beautiful tree ferns. This was like 180 million years ago about. But also what most people don't know, and definitely people that did the Jurassic Park didn't know, is that the diversification of the macrofungi, the Basidiomycota, also happened in the Jurassic. So this is a graphic, an image from this paper from 2019 that shows like when the different forms or body forms starting to appear. So here we have like fungi that didn't form any type of basidioms. In orange, we have all the resupinates, the fungi that grow like, that they look like a ink in a wood or something, which were like dominated for a long time. But then when we get to the Jurassic, we start seeing all these different colors in the graphic that are equivalent to the forms that are here. So pilate fungi, cephaloid, gasteroid, and coralloid fungi, they all started to diversify and appear during the Jurassic. So that means that everything that we have about the Jurassic is or representing the fungi, or not even representing the fungi. So I think there should be another Jurassic Park movie where they would show like the dinosaurs eating mushrooms. This is only on my head, but I mean, there are no evidences about this, but I think that they probably did. I mean, it's a source of protein, right? If you're a dinosaur and there are all these beautiful fungi, wouldn't you eat it? Well, then from the Cretaceous, we have what is known today as the only mushroom fossil that is preserved on rock. Because all the other mushrooms, or the other fossils were generally like associated with the wood or plant cells. And it's really rare because the fungi are so ephemeral that it's rare to see a fungi or hyphae that are preserved on rock. And this was the first one that was found. It's from Brazil, actually from northeastern Brazil, and dated from the Cretaceous. So you can see here like the shape of the mushroom. This is about it's like about a centimeter tall. And this was found in Bacia do Araripe. It's a part in Brazil that's famous for several fossils from different origins. And it was actually sent to the US. It was studied by heads and collaborators at the Field Museum in Chicago, and then now it's back to Brazil, and it's like in a fungarium in the northeastern Brazil. Then in contrast, there are several fossils of mushrooms that were kept in amber. So that's a little more common because the amber envelops the mushroom when the mushroom's still like fresh, and then it solidifies and it forms a fossil involved in the amber. They look really pretty, right? And they look just like mushrooms that we can find if we go outside right now, like in our yard. So here I brought two examples, one from the Dominican Republic. This one is from Myanmar. And these are mostly, there are several other examples, and these are mostly from the Cenozoic, like 66 to 15 million years ago. And this, also from the Cenozoic, there's another mushroom on rock that is discovered, that was made in Puebla in southern Mexico, and it's an earth star. And I mean, it's unquestionable, right? Because we see all like the rays from the exoperidium and the endoperidium in the middle. And this was also preserved on rock. And it's called Geastrum tepexensis. So we have all these forms that we find today that are very similar to the forms that were diversified since the Jurassic. And so here I have like an abstract, like right, with all the points where we saw these events. So from the Proterozoic to the Silurian, and all the way up to like the Cenozoic. And I think this is fascinating because it's like they had a big impact. They still do. And I think we have to rethink how we imagine things. So I brought the same image here with like some fungi among the dinosaurs and the tree ferns. And I hope this is like interesting and helpful, and I hope you have enjoyed. And if you have any questions, or if you want to look at any of the articles, you can just ask me. I can send you the articles later. Also, the list of references that we used to prepare this talk and to make this chart is on the Portuguese version of the booklet that I showed to you. So with that, I would like to thank you for the time. Thank you, everybody that came to listen to me. And if you have any questions, I'll be here. Thank you so much.
Hey, thank you, Maria.
Thank you.
Jan, have you been watching the chat? I haven't really
I have
been.
been watching the chat. There was just one question,
No.
but I'm gonna look right now if there's any other question posted on the main chat. Someone says thank you. I think it was a
These
great
are my
presentation.
contacts also, if you
Oh, yes. Do you want to put them in if you see the chat, you want to put them in there? Oh, you have them right here. Oh my God. Okay.
okay.
I
But I
posted
can
your
put them in the chat.
Yeah, no, it's fine. I can do that. I post them I post the Instagram on the chat, and also the book How I Found You, right? The Let's Become Fungal.
Uh-huh.
I had a question about the very first one, the oldest one. It looked like it was maybe in the water or something. Where was that from?
Mm-hmm.
Do you remember? If you go to
No, I don't remember.
the
Yeah,
very,
like
very
this one.
first,
Oh, yeah,
It
this
looked
one.
like brown
that
balls.
one,
There.
this one. Yeah.
Or was
You,
5 years old. I don't remember. I would have to look on
okay,
the the um, the paper.
the
It's
paper.
not
Okay.
very old paper, as you see. It's from
I
2021.
see that. Yes.
Yeah.
Yes. I actually looked up some stuff too, and like the oldest ones were not this old, the ones I found. I know maybe in 2021 there was something that was found in like a Congo somewhere.
Mm-hmm.
And that was supposed to be one of the oldest fungi that was kind of between the earth and the water, like when the plants were kind of like maybe migrating to the earth, right? I
Uh-huh.
mean, to the dry land, sorry. Um, Here's a question. What is the name of the book you mentioned at the beginning of your talk? I did post it, but I'm going to post it again.
Okay.
Let's Become Fungal. I just finished reading it. So I'm going to post that there again. But did someone else mention a book? I thought someone else mentioned some other book. No? Oh, that was Susan. Sorry. Susan had another book for us. So I'm posting
All
it
right, at the
again.
before we started, yeah.
Yes. So I'm not sure if anyone
I
has
can
any other
put my
questions.
book in the chat.
Oh, yes, yes.
That sounds
Maybe,
great. Yeah.
um I had a question about the decomposition, like the brown rot. We call it cubicle rot too, like in the club,
Oh,
right?
okay.
Right? Is
Uh-huh.
that David? Is that like the official name or no? I don't know.
I don't know.
Makes sense.
Everybody says brown rot, white rot. You know, that's
Yeah,
the
brown rot,
traditional
white
scale.
rot. So, uh, is it true? I think so that certain fungi only decompose using the well, they only turn it into the brown rot, and the other ones will just turn it into the white rot, right?
Mm-hmm. Yeah, what happens is that eventually, even with the brown rot, some white rot fungi will take over and finish the wood, like
Okay.
decompose the lignin. So what happens is that the brown rot can only access the cellulose, but then eventually some white that doesn't stay that forever. So some white rot fungi in the sequence will grow on that wood and decompose the lignin
Oh, okay. I
and
didn't
do
know that.
that.
Okay,
Yeah,
I thought it was one or the
yeah.
other. Like it cannot be one or
No, yeah, they can grow together, but if the brown rot grows first, then the wood looks like the cubes.
yeah.
But then eventually that is also be decomposed by a white rot.
Mm-hmm. Oh, I didn't know that. That's
Mm-hmm.
something new to me. I was wondering about that.
Yeah.
I gotta say, Maria, it's unrelated to kind of most what you've been talking about, but I have never seen Lacaria lacata look like that. Those are really cool looking mushrooms. It must be completely different than what we have up here for that species.
Well, that is actually not my photo. It's not from Brazil because we don't have lacarias in
Yeah.
Brazil.
Ours like
We,
nothing like that. Those are
yeah.
really neat looking mushrooms.
Yeah, I like to follow up with the Lacaria lacata as well. So it is toxic to the Colambula species.
Uh-huh.
And it picks up the nitrogen and then passes that on to the Pinus species. Um,
Right.
yeah, this is fascinating. Is it species specific? Is it just the colambula that the Lacaria attacks?
So this specific word, this is an article from 2006. It's pretty old, and it's the only one I know that of this kind. So they were testing one species of colembula with several species of fungi, and the only one that gave that result was lacata. So we actually don't know. I think that it's a very hard study to know because you have to happen to find the exact match, the exact pair that works like that, you know. So with so many, I don't know how many species of Colembor are there, but there are so many like insects, right? Insects are hard. So you'd have to just happen to find the same, or I mean, the species that match. And my thinking is that if somebody found that one relationship, it's probably because it happens more often. But so that was made with only one species of Colembla.
Now, is this the similar process to what happens with oyster mushrooms when they're eating nematodes?
Yeah, I think so, but I don't know because they are not mycorrhizae, so we don't have the fact that they are transferring to
Oh, right,
another
right, yeah.
plant. But yeah, I mean, we have fungi using animals also as food sources a lot. So that's why I think that that might happen. So I always joke, I have like students that are vegans, and I joke like, you don't know if you're not eating animal nitrogen in the plants because of the fungi.
Right. But I think one difference between, that all oyster species feed on nematodes. It's like diagnostic for that whole group of mushrooms. But in the case of the Leucaria and the Colembala, I think it was, you pointed out that it was that particular species of Leucaria and that particular species of Colembala, because I only know of one other study that looked at this afterwards, which was done in Scotland with different fungus and different Leucaria, and it didn't poison the Colembala. So it may have been that Chloronymus, who did that original study, was just lucky in those species.
Exactly. And that's what happens in science. Sometimes you have a question and you are looking for something, and just by chance you find the answer to something else that you had never even not even thought about it. And that's very common, but you can only see that if you are alert and if you are like, because I think a lot of times what we academics do is we ignore what is not within what we expect. And then we miss a lot of data. I mean, I'm not saying everybody, but I think we have to be open like to results that are unexpected. And I think it is a little luck sometimes, yeah. And also with the oyster mushroom, we know more because it's a more commercial fungus that have more economic interests, right? So there are more studies. And that's one thing in science that when we have questions that are not economically important, it's harder to get funds, it's harder to pursue with the project. So who cares about Lacaria eating colembala, right? I mean, what does that say? I mean, it says a lot to us that are curious about fungi and the interactions, but um, it's complicated.
Maria, when do you anticipate the translation of your book, the one in Portuguese into English, if I may ask?
Oh, I don't know. I've been wanting to do for a long time. Maybe this talk will give me the the start that I need. I don't know yet. I'm at the end of the semester now, but I hope soon. I hope I can do it soon. It's more about like time.
Thank you.
Sorry, but I can send it to David and Jenna, and they can share it with you when I have it.
That would be wonderful. Thank you.
That would be wonderful too. Me as a teacher in school, middle school, I think it will be received well because there's not much resources, right, for people to include the fungi in a curriculum, and um, even for the club, I think it would be great to have that, right?
Yeah,
You want
absolutely.
to share that with the BMC, that would be wonderful.
Yeah, I
I
will.
think that would be great whenever that happens.
Okay.
So you think maybe next week?
Next, yes,
Oh,
by the end of next week.
exactly.
Yes, 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 yes. Oh, I want to also, if no one has other questions, I want to mention I posted the Spun link at the top of the chat, but I want to say that Maria also work with you did a research with them, a grant or something, right?
Yeah.
Society for the Protection of the Underground, the Conservation of the Underground. So it's kind of like like the citizens scientific research, right? When people go and take samples, I guess from all
Mm-hmm.
over the world, right? You would know more. They take samples of the earth and then they send it somewhere, and that is being really looked at and analyzed what's in the soil, right? And
Mm-hmm.
you said you did that for some areas on your island, and I think anyone can, I guess, take part. I'm not sure who you have to be to do that.
Mm-hmm.
So,
Yeah, so if I can talk a little bit about that. So what
yes.
I did, they have like grants, so I applied for a grant to look what ectomycorrhizae, because I have a lot of data of mushrooms that form ectomycorrhizae. Here I am in the map. Can you see the map? So we have I have been collecting mushrooms and ectomycorrhizal mushrooms and other mushrooms in the island like for a long time. So we have the information of the mushrooms, but there's nothing really or nearly nothing being done in the neotropics actually as a whole with ectomycorrhizae, because there was like this myth that ectomycorrhizae were not present in the tropics, but they are. But there are just like not enough studies. So we have been collecting and studying the ectomycorrhizae and trying to find the match, trying to find like which are the plant hosts for the ectomycorrhizae that we have here. And that's a hard work because we have to dig, we have to find the root tips, and then we have to sequence because we have like such a diverse forest that it's impossible like to know which are the root tips that we have to match with the trees. So we do like what I call like a parental molecular study. So we have the root tip, we sequence the mycelium from the mantle, and we sequence the root itself to see who is the mushroom and who is the plant. So we were doing that, and then the SPAN project came along, and my goal was to compare what we had from the root tips, from the mushrooms themselves, with what's in the soil, or meaning the mycelium. So I proposed a project that we collect in areas in the south of the island. So we collect here in forests, in areas with dunes that are called restinga. These areas have a lot of mycorrhizae because the soil is sandy and very poor in nutrients, so the plants need the mushrooms. And also in mangroves. And then we did the same thing in an area like more like in the middle of the island, and then in the north part of the island. So we finished the sequencing. We have a lot of data now because basically we get a sample of soil from different points, and we sequence, and we have a list. We use like a specific primer, specific region to help us identify the Basidiomycota. And then with that, we have a list of species that are in that sample that we can then compare with what we have in the database for mushrooms and for roots. So it's really cool. I haven't worked with the data yet, but we have data for a lot of projects now. So we got the money to do the sequencing, to do the field trips and to do the sequencing. And now we have all of this data to work. So we give all the data to SPAN, to the society, but we have all the data like to work and do whatever projects we want from now on. So it's cool. And one of the ideas that we have is exactly to compare what's on the soil, below soil, and what we have found on the top. But I still don't have like that those data analyzed.
I just say
And
hopefully by
mangroves.
next week, translation, you'll have that.
Absolutely.
Yes, yes. I'm not sure if anyone else has any other questions,
Yeah,
Susan, or
I
anyone else,
just have
David.
one last question. I can't
Yes.
pass up the prototaxites. I mean, why did it get so big?
Oh.
Any reasoning? Was it the atmosphere that contributed to its size?
I don't know the answer to that. I'm sorry.
Mm.
There's
I
a
don't
lack
know.
of mycologists eating it.
Yeah.
26
Maybe.
feet long. I mean, that would be a banquet.
Yeah, right?
Yeah.
Interesting.
Thank you. It was a wonderful talk. Thoroughly enjoyable.
Oh, I'm glad you liked it.
Yes, we hooray
Um,
for you. Thank you.
Well, thank you very much. Thank you for watching.
Thank you coming all the way up to North America for the
Yes.
lecture. It's a
Oh,
big
I wish
journey,
it
but yeah.
it's so cold here.
Yeah. Yeah, we're in just a brutal heat wave, so everybody's kind of a little jealous of when you say
Yeah.
it's
Yeah,
I
love it.
know. Well, send me an email or find me on
Yeah.
Instagram if you have any other questions. I'll be happy to answer.
And I'll send you a copy of the recording when I get it from Zoom.
Perfect. My
Okay.
students were curious.
Yeah.
Thank you very much for attending, everyone. Thank you.
Yeah, and I want to thank Yana for doing all the legwork to track down Maria and get this all set up. So it's always good. That's anybody volunteers for anything, I'm happy. All right. Well, thank you
Thank you
very
so
much.
much.
It was great meeting you.
Great
You
meeting you,
too.
Maria. Have a great evening. Goodbye.
Okay,
Stay
good night,
warm.
You
everybody.
too.
Thank you.
Goodbye.
Bye.
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 Stay cool.
Yeah.